Okay, super. Well, thank you. That's a very kind introduction. It's a real treat to be here. I've been looking forward to this seminar visit all semester, and so it's nice to finally be here, meet many of the new people that I didn't know before, and then put some names and faces together as well. It's also very lovely. So I was asked to talk a little bit about sort of open data science and some of the projects we're doing in database design and development. And so what I'm going to do is I'm going to spend the first half of the talk talking about some of the platforms that we've been involved in creating and maintaining. And then in the second half, I'll tell you one application of how we have used those data in order to develop a new tool for natural products discovery. So before I get into the details, I thought it would be worth summarizing in more general terms what it is we're interested in. So I'm really a true natural products chemist. So we're interested in these two core questions in natural product science. The first of which asks, what is the chemical diversity of the natural world? This is obviously an ambitious and sort of career-long question to try and answer. And this involves some analytical chemistry questions, as well as sort of databases, interacting with colleagues who study genomics and metagenomics, et cetera. It's a sort of broad picture question to try and resolve. And then on a more practical note, how might we apply those molecules to problems in human health? And so we also think a lot about high-throughput screening, biological properties of some of the molecules that we have discovered or that are available in the environment to study. And so anyone who's done anything in the sort of natural product workflow is likely to recognize this cartoon slide. This workflow hasn't changed very much in a very long time. So you could argue that this is the same workflow by which penicillin was discovered and many other compounds that we take for granted as drugs today. So you typically, you start with, let's get my pointer, there we go. You start with some source organism. In our case, that's environmental microorganisms, but it might be plants or sea sponges or higher organisms. You'll take a library of these organisms and convert them to extracts, and these are typically mixtures of components. So these are either just crude extracts of the organism itself, or sometimes some kind of blunt pre-fractionation step to set them up as libraries. And then those libraries are subjected to both some analytical methodology to try and understand the chemical composition of the libraries, and then some screening methodology, here a cell-based screen, to tell you something about their biological function. And then once those two pieces of data are in hand, one must then integrate those two data sets together into a single data set, which hopefully will allow you to draw connections between presence or absence of chemotypes in your data set and presence or absence of phenotypes, with the long-term goal then of selecting those molecules most worthy of development and eliminating the things which are perhaps aspects of rediscovery or molecules which are known to have liabilities that don't make them useful for your biological endpoint, whatever that endpoint is. And so when I first started as an academic, we did this in very much a manual way. So we would collect small numbers of samples and we would get some screening results, and each student would get a sample. I mean, this is how I did my PhD. It's how our early projects were in the lab. And it has been an extremely effective way to find drugs. But even when I was a PhD student in the early 2000s, I isolated 19 known molecules in a row before I found my first new molecule. And one of the molecules I isolated was published by my PhD supervisor when he was a graduate student in the 1970s. This is not very efficient. So we became interested in sort of systems approaches. So rather than studying the chemistry at the end, where you pick your extract of choice and then you study the chemistry and you work iteratively through to identify molecules, why not look at a much broader scale of the chemistry of the set at the beginning, and then try and draw inference about which molecules are worthy of your attention sort of early in the process, so that you really focus that dedicated and complicated chemical effort on molecules most likely to be of value. And so that led to us kind of reversing a lot of the workflows that we pursued, and it meant that we started to study samples on scale. So a typical experiment might involve 1,000 extracts. And as soon as you start doing that, then you start needing informatics, because those data sets become big very quickly. They become really hard to handle and very difficult to interrogate. And that led us to start working in the area of sort of informatic tool development. So in the last 5 years, we built a number of open source tools which aim to address bottlenecks in various places in this discovery pipeline. So I'm not going to talk about most of these today, but I will just quickly summarize what they are and what they do. So they're almost all published now. at the beginning, multiplex MS. This is a tool for accelerating the number of samples you can acquire untargeted metabolomics on in a single experiment, designed to increase throughput by 30-fold over traditional replicate-based methods. MS2Analyte is the only tool that's not yet published. This is a data processing package for untargeted metabolomics data. It's a vendor-independent mass spec data processing package, which I'm still trying to finish. It's been a long-term project for me. NP-Analyst is a tool that does data integration between untargeted metabolomics and screening data, aiming to make those direct correlations between phenotypes and in this case, MS features. Then the last three on the end here, these are the ones we're going to talk about today. So NP-Atlas is a database that contains all the known microbial natural products that have ever been published, or at least that's its aim. SnapMS is a tool for reference library free annotation of metabolomics data for natural product structures. So it aims to essentially use the information that's in the Atlas database for inferring chemotypes and structural diversity in large data sets. And then the NPMRD is a collaborative project with other colleagues in the US aiming to build a database of NMR data, so nuclear magnetic resonance data for natural products. So what I'm going to do today is I'm going to focus on the N part of this pipeline. So I'm going to tell you a bit about sort of database development in these two areas. First the Atlas and then the NPMRD project. And then I'll tell you a little about SnapMS and how it works and how it uses the Atlas as its frame of reference. But one does not grow up wanting to be a database administrator. So I didn't think that would ever be me. And so I thought it would be useful to tell you sort of why I ended up doing that. So really this came about from a paper that we did with other colleagues in San Diego and Santa Cruz back in 2017. And what we did in this paper is we took a commercial database of all the known microbial and marine natural products that had been published up to 2012. And we augmented it with a few more years, like 3 more years worth of data that we extracted manually from the literature. And then we looked at how rates of novel natural products chemistry discovery had changed as a function of time. So this plot, the red line shows you the number of compounds per year published in the database. So in the very early years, in the early 1940s, oops, I'm pressing the wrong button, let's try this one. There we go, come on. So here in the early 1940s, the number of compounds found per year was really low. And that was in large part because analytical methodologies were not very good. Many of the tools we take for granted didn't exist. And determining structures was really, really difficult. And so consequently, it was very slow. There was also nothing known. So every structure was a truly de novo structure determination effort. And that made life very difficult. And then as you go through the 50s and 60s into the golden age of antibiotic discovery, and then up into the sort of early mid-70s, then the number of compounds discovered per year starts to rise. Does anyone know when the first 2D NMR experiment was published? It was 1984 for the HMBC and 1986 for the HMQC. So for those who use NMR for structural elucidation, those are critical experiments. And so as soon as those experiments became kind of mainstream, you can see a concomitant sort of rise in the number of structures that get solved per year. It's not just NMR. There were improvements in mass spectrometry. There were lots of improvements in other areas, HPLC, lots of other tools, but they definitely contributed. And so by the late 1990s, the number of molecules per year is about 1,500, and that sort of stays fairly consistent from then on. But the blue line shows you it's a plot which tells you about the percentage of compounds. So let me step back and explain how we did this. We took all the compounds in a given year, and we used a structure similarity scoring metric to assess how similar they were to compounds found in previous years. And that score goes between 0 and 1, where 0 is no atoms in common, and 1 is essentially overlaid, superimposable molecules. And so we set an arbitrary cutoff of 0.4, which is a pretty low number. And we asked what % of molecules found in a given year were really unusual compared to stuff found before. And what you find is that at the beginning, everything is unusual because there's nothing in the data set, and basically everything anyone touched was a new class. And then as time goes on, you see the percentage of molecules which have got very unusual scaffolds sort of goes down and down. And some in natural products don't like this plot very much because they suggest that it offers a sort of death knell for the field and that everything there is to find has been found. But of course, this is a percentage, and the number of compounds per year being found has gone up a lot in that same time. So if you look at it as an absolute plot, what you find is that actually the number of compounds that are really unusual has risen since the 1940s and has stayed pretty constant, notwithstanding the fact that as a field, we add 1,500 new molecules to the pool of known molecules year on year on year. So this suggests that, within the available space for natural products, there are a surprising number of really strange and unusual structures to be found, and we found them at a reasonably consistent rate. So it suggests that we're not depleting the pool of structures that are known out there. But it does suggest that if your goal is to find structurally unique aspects of chemistry, you really need to think critically about your discovery modality. Because if you just blindly go into the forest here and you pick some streptomyces, the odds of taking 10 streptomyces and finding a new natural product are pretty low. So people sometimes ask what these are, so I picked out a few. So these are all molecules that score highly on the unusual chemistry index. Some of them you can sort of imagine. So this aziridine-containing natural product is relatively rare. Even this chloropyrrole is a reasonably unusual functional group. But some of them, like this one, I'm slightly surprised that this is in the pool. It has a lot of aspects which are sort of reminiscent of lots of other natural products. But obviously the architecture, the assembly, the ordering of those subunits must be very unusual in some way for it to have ended up on this list. So it's an interesting exercise, and it kind of fueled my interest in understanding at a broad level what's going on in the natural products world. And so that was why we became interested in this project of database development, and it's what led us to lead a project to develop the Natural Products Atlas. So, as I said, the Natural Products Atlas, it's a free and totally open source database which aims to include all the known microbial natural products that have ever been published. It turns out that that's quite a hard set to encompass, especially the older papers are quite hard to curate, and I'm going to tell you a little bit about that process in a minute here. We didn't do this on our own. I should say up front that although our lab maintains the atlas, it's built with the help of a huge array of collaborators, and there's a slide at the end there which tries to list all the people who've made substantive contributions. If you're interested in this topic and you would like to help, you're always on the lookout for extra volunteers, so you're very welcome to join in. We tried to set the tool up to be as flexible as possible. So you can download the dataset in a variety of different formats. There's a reasonably accessible website that allows you to search the data in a bunch of different ways and provides some different data visualizations. And if you're a programmer and you want programmatic access, there's also a full API on the back end which allows you to do like system-to-system connections and cross ties and things like that. And I'll tell you a little bit about that too. But obviously, if you just looked at this as a project proposal, like a grant proposal, which said, we're going to read every paper anyone's ever written, and if it's about natural products, we're going to get the natural products data out of it, you would think this is a completely insane proposal, like it would never get funded, because that's an impossibly difficult task. But actually, it turns out that the literature about natural products is readily recognizable. So this slide here, it's got a lot of words on it. But what I did here is I went to the year 2010, and I went to 3 journals that publish a lot of papers about natural products. And I started with the first paper in January 1st, and I read through all the papers until I found the first one that was about natural products, and I copied the abstract and put it on the slide. And if you look at the 3 abstracts, I've highlighted a few phrases, like in the different colors. Actually, although they're about completely different projects with completely different structures, it wouldn't take very much to recognize that these are all about the same kind of subject. And in fact, you could train high school students to go through papers and say yes, no, yes, no, yes, no, and they'd probably be pretty good at it. So this is a really great situation for a very simple classifier model. And so what we did is we took the initial work that we had done curating some years worth of data, and we paired that with some more manual review of all the articles in journals we knew to be relevant to the natural product space. And we built a matched training set of 20,000 articles. half of which were about natural products, half of which were not. And then we turn them into a simple classifier, and now we just run the classifier over everything that's ever been published, and we find all the papers that are about natural products. And that's how it works. So in terms of how we build the database, originally we had a huge push where we recruited 40 volunteers to help us. And we picked out 50 journals we knew were about natural products type articles, and we ran the classifier over everything they'd ever produced. And then we got people's help to curate those data. I'll show you how in a second. Nowadays, it's a little bit easier. We don't need quite so many volunteers because we've covered a lot of the historical data, and we're mostly focused on the current and future papers. But the workflow is always the same. So we always start with a pool of primary literature, and then we use this SVM model to pick from that literature just those articles which describe natural products discovery. And then we have a second set of tools. So this NLP stands for natural language processing. So we have a set of tools here which look in more detail at the text of the abstract, and they recognize genus and species terms for all the organisms. They recognize compound name terms for all the compound names. And they deconvolute situations where compounds might be listed in groups. So a lot of these, well, I can show you actually. I wonder if I can. Oh yes, I can. Good. So here, there are 3 compounds, all of which are listed in a sort of block of text here. So the system will recognize that they represent 3 compounds, and it will pick them out and list them as A, B, and C separately in order to get a proper picture of the chemistry that's in that article. So then we know a lot then about which information are present in each article. The only thing we don't know is the structure. If we have the name and the compound and the paper is old, we can go to PubChem or Chembl or other open databases and get the structure. If the paper is brand new, oftentimes we have to enter them. Which is a bit tedious. And so we pre-format all the data ourselves internally, and then we recruit curators to go through those data and check them and make sure they're right, make any corrections, fix structures, change names, sort out misannotations of species and things like this. And then we undergo a set of about 50 quality control and standardization checks before we take those curated data and we insert them into the database. So we want to make sure that we're not inserting a compound we already have. We want to make sure that the genus and species terms are recognized by some of the standard databases of taxonomy. We want to make sure that the compound name hasn't been used before, that you're not polluting the database with cross-naming. There are all sorts of pollutions in the literature. You find crazy stuff. The same name used for lots of compounds, the same compound with lots of different names, all sorts of strange things happen. So we do a lot of checks to try and make sure that the data are as clean as we can manage. And then once they're inserted into the database, before we release a new version, we then go and make cross-links to a lot of other data resources. So some of them, LPSN and Mycobank, these are taxonomic databases. Unichem is a sort of aggregation service for connecting to Chembl and other chemistry data resources. NP Classifier is a tool out of San Diego that predicts the biosynthetic class of each molecule, so that you also have definite, is it a terpene or an alkaloid or glycosylated polyketide, etc. NPMRD is our NMR database. You're going to hear more about that in a minute. GNPS is a database of mass spectral data for natural products. I'll tell you a little bit more about that later as well. And then MIBIG is a database of biosynthetic gene clusters for natural products. And so we make bidirectional crosslinks with all of those resources, and we append those to the entries in the database. How do curators actually manage to curate all these data? Well, to do that, we built another full stack app, which is not public. So you only get access to this if you're on our curation team list. We'll give you a login and set you up in the system. So we pre-process all the data and put it into sets, and then we assign the sets to curators. And this is an interactive web page here. You can review the data for the citation and various other details about the structures. You can click through, so there's a depending on how many compounds there'll be a list of tabs here. And if the compounds aren't right, you can replace the smile string, so you can update the structures so that the structures you include in the database are accurate, etc. There's a bunch of like housekeeping that you can do here. If they're really unmanageable, you can check the needs work box. This is where, to be truthful, this is where papers go to die, because usually it means there's something very strange about the paper. Either it's not really clear whether it's relevant to our database, or there's something really difficult about the way the structures are being presented. And I'm supposed to go and look at these special cases and sort them out, but they're always really time consuming, so it tends to be difficult to keep up with them. So we try and encourage people not to do too much of that if they don't have to. So anyway, they go through these data, and once they've done it by hand and looked at every entry, then we use the automated checks and we insert everything into the database. And so oddly, it's a database that doesn't actually have that much data in it. So it has citations and structures and names, but mostly its value comes from its cross connections to other resources. Because there are a lot of specialty databases out there which cover natural products in one way or another. But if I have an example here. So if, let's say that you are a researcher who's interested in this compound class. I picked it sort of arbitrarily out of the data set here. So one of the things the atlas does is it gives representations of sets of compounds that are similar. So we call this a cluster. So every node in this network is a different compound. But they're all structurally related using that same structure relation methodology I described before. If you look at the NPA ID number is a rough chronology of when it was curated. It doesn't tell you when it was published necessarily, but it does tell you when we inserted it into the database. And sometimes if the molecules are similar to one another, it's because there were 5 compounds in one paper and they're all there together and you can see the numbers are sequential. But here you can see that actually the numbers aren't sequential at all. Like some of them, this is 82, is one of the first ones we ever put in there. And this is 31,000, which was done very comparatively recently. So they're probably from different papers of different vintages, different variants of the same scaffold found by different people. So the database is quite useful in that way, in that it tells you, okay, if I care about this core, what other things are known that are in this general chemical space? But the other things that are quite useful are that if you have information about these clusters, you can also look at where the crosslinks are. So although you might care about this molecule right here, if you look at the biosynthetic gene cluster database, you might find that actually there are several molecules in this set which all have some kind of biosynthetic gene cluster information, which might be useful. It may all be the same VGC, or it may be that there are various, you don't know. You can go to MIB and check out and see what's there. If you want reference MSMS data, 2 of the members in this cluster have reference MSMS data in GMPS. So that might also be useful for doing annotation and cross-comparison. And then if you want NMR data, some subset of these have either experimental or calculated NMR data that go with them as well. And so that kind of information is actually quite hard as an individual user to sort of gather together. And so one of the big values that we see in the database is that people can use it for discovery in lots of ways like this, which I think is kind of interesting. And actually people so a lot of people asked when we started, they said, oh, it's really kind of you, but like, why would you do that? It's going to be hugely time consuming, and you're going to get one paper which says, oh, here's a database we made, and then you're going to be stuck curating this database forevermore. Like, this is a terrible return on your investment. And it's not entirely self-serving, because of course, if you become known to know about that subject area, then people come to you with problems and they bring new and interesting questions to you that you hadn't really thought about before. So it's been quite a fun way to build new collaborations with people and like meet new people and do new projects. But it's also been really interesting to see how the wider scientific community has used the databases in ways that I didn't really expect. So I thought people would use it for dereplication. I thought they would use it to sort out which molecules they had amongst their set that were new and publish new molecules. But it turns out that that's not true. Well, it is true partly, but not completely. Let's see if I can see my notes here. Yeah, so we've published 2 papers on this. And as of yesterday, there are 319 citations. And approximately a third of them do dereplication. They say it's a paper which is about finding some new molecules. And it says in the workflow, as part of our early workflow, we used the atlas to do some, to see if this was a new molecule or not. About a third of them are review articles, which say, mostly they say natural products are really great. Look, here's a bunch of natural products. But then a third of them are new tool development, which uses the atlas as its reference set for the development of new tools and methods. And that, maybe it was naive, but that was unexpected to me when we started out. I thought the number of people who would really use the data set in that way to build new tools would be relatively low. But actually, it's really much higher than you would think, which suggests that if you want to find new chemistry, and if you want to sort of advance the field in that way, and you need new technologies, the thing that's missing for the field at large to develop new technologies is high-quality reference data sets, which I think is an interesting prospect. I think that the funding agencies are catching up with that idea. Until quite recently, funding for database development was extremely difficult to come by. It was considered a sort of chore. I don't think that's so true anymore, but I do think it's important. I think they are, they're useful resources, sort of broadly writ for the community. And I think if we as natural products chemists want to stay relevant, you can't keep doing things in the way you had done before. You have to sort of like find new ways of solving existing problems, and this is one way that you might do that. Okay, so that's the Natural Products Atlas. So one of the things for our lab that came out of that is that 2 years or maybe a year after we published the first paper on the atlas, the NIH put out a call for groups to develop a database of NMR data for natural products. And so we submitted a proposal to this program, and we were fortunate enough to get funded. And so we started this project called the Natural Products Magnetic Resonance Database. So this is not just me, this is a consortium project. The other team members are down here in the bottom right corner. Here we go. And together, this group has built a database which aims to gather as much NMR data as possible for natural products. So NMR data is almost always used for solving structures in the natural products world, but it's almost always discarded in the publication process. Typically, those data are distributed either as a picture, PDF in the supporting information, or as an assignment table. So maybe there's a table with a list of the numbered positions of the carbons, and then the shifts, carbon and proton shifts. Or some, rarely now, but sometimes just as a list of shifts. I got this spectrum and it had these shifts in it. But the information content in the original raw data is actually really high, much higher than you would think. And those data are largely lost to history, like they're sort of gone. Before I started grad school, they used to save the data on 8-inch floppy disks. Anyone remembers those? My thesis was on a zip disk. Anyone remember those? Magnetic tape was quite popular. And then early like Unix and Windows systems, and like almost all those old storage systems are extremely hard to access now. It's really difficult to get legacy machines up that will actually read those files. And when you do read those files, they're probably coded with the code names from the lab book of whoever acquired the data and whenever they did that. So it's effectively impossible to go back, even for people who have those disks in their storerooms. It would be really difficult to turn those disks into curated NMR data. When is the day that people are most excited about any science project? I can think of two. It's not rhetorical. You're supposed to answer. So when are people most excited about their project?
When it gets funded and when it gets published.
Yeah, I would say definitely when it gets published. I would say when it gets conceived. Like that first day when you got a bit of paper and you sort of, you draw out ideas and you talk together. That one's fun. Funded is, I guess there are three. But definitely when it gets published. So we recognize that if we wanted to collect NMR data, there were really only two ways to do it. One is to go by hand back through the literature and transcribe the tables of data and structures back into a machine-readable format. And we've done that. So we built a system to do that. We hired people to do it. We've done about 4,000 molecules in that way. It's extremely time-consuming. Even though we built an app to help us do it, it takes ages. It's really a tedious manual process. So the only other way to do that is to encourage the community to put that data in on the fly. And in the long term, it's the only practical way that we'll have a reasonable enough data set. So our program then is really cut into two halves. One part of the team houses the database, and it runs the website that allows you to access the database. And that's run largely out of Alberta by David Wishart. And then the other team does the collection of the data. So we built a totally separate stack which helps people with like depositing and managing and curating data. I'm going to tell you a little bit about that here as well. What we did is we built a website with a database behind it, which allows people to drag and drop their data and deposit their data very easily. And I'll show you a screencast of that in a second. And then we also built a system which tracks the literature in real time. So we recycled some of the tools I've told you about already. We built a system that tracks the RSS feeds from the 50 journals we know publish most articles about natural products. And we use that same article categorization tool to find the articles that are about natural products discovery. We then use the natural language processing tool to pick out the source organisms and the names of all the molecules, etc. So that's ML model to find which papers we care about, the named entity recognition for getting the information from the abstracts. And then this information goes to an in-house data scientist. At the moment, that's Ella Poynton from our lab. And she looks at every one of those automatically generated summaries, and she fixes any problems and sorts out typographic mistakes and missing compounds and things. She also identifies the surname of the corresponding author and their email address. And then we put those correctly. I don't know how many of you get emails addressed to your former graduate students and things asking for papers. I get that a lot. So we try really hard to make sure that they're right. And then we email the corresponding author on the day the paper comes out. And we say, we really like your paper. We'd love it if you put your NMR data in our database. We've made a personalized page for you that contains all the information we know about your paper. So this is what happens if you go to that page. So you get straight to a page which names the compounds. It says what genus and species they came from. It typically doesn't have the smiles pre-filled because that's the only detail that we can't get easily from the abstract. So we ask people to put the smile strings in and then to zip their NMR data and drag and drop it into the box. I actually have, let's see if we can get this to run. So I have a little video about this. Come on then. Here we go. So we ask people to paste the smiles. So what I'm doing here is I'm going to ChemDraw and copying the smiles and then coming back, pasting it in the box. And then I'm going to go off the page again and get my zip file of my NMR data. I'm going to drag and drop that into the landing zone, do it for both compounds. And then we're going to click validate. And the system's going to think about it for a bit and inspect what's being deposited. I'll tell you about that in a second. And then it gives a summary of the structure, a picture of the structure, a list of all the spectra that you deposited. If you like it, you're done. So the goal was to allow people to do it in under 5 minutes. What do you think our response rate is?
10%.
Oh, you're really good. It's 8%. Which is a little bit disappointing. I mean, I sort of see why. There's a lot of deposition fatigue. Any of you who've actually uploaded papers for publication, it's really tedious. There's loads of form filling to do. You've got to find the middle initials and the email addresses of every author. You've got to make sure they're in the right order. You've got to find the contact details for the referees you want. It goes on and on. So the idea that it might be finished and then someone would send you another form to fill in, enthusiasm is understandably low. Encouraging researchers to think about the value that adds broadly to the field is really important. And that's done partly by sort of incentivizing people to put the data in by giving them tools and getting value add from putting the data in there in the first place. Maybe better citations, maybe you get a DOI for every spectrum you put in there, things like this. Or it's by mandate, either by funders mandating it or by journals mandating it. And both things are starting to happen. The only way that we can do that 5-minute deposition is by doing a lot of work behind the scenes. So if you look at, so what I have here is a workflow. So in blue here on the left, those are the steps I just showed you with the mouse, click, 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 going through. But actually at every step, behind the scenes, the back end of the database is doing quite a lot of work. So when you first input your structures, the system will validate the structures. It will first ask, is this a valid structure at all? Like, is it in the right format and is it reasonable? Are there any like pentavalent carbons or other like crazy errors that we need to fix? And if there are problems like that, it tries to give high quality error reporting. So we have a big long list of ways in which things can be inappropriate. And for each one, we try and give customized error reporting back so that if you're a user, you have some chance of knowing what the problem is and trying to sort it out. The NMR data is actually a really complicated thing to distribute, because in the other database, all we distribute are like citation information and smile strings of structures. It's all text. So it's really, from a security standpoint, it's really safe. But NMR data are directories full of raw data. So from a security standpoint, they are much less safe. If someone gives you a zip file and you take it on trust that it's NMR data, but it's actually malware, that would be very problematic. So we have to be much more careful. So when they upload the NMR data, we ask all sorts of stuff like this, like, does the zip contain malware? Does it contain any NMR data? You'll be surprised that sometimes the answer is no. Or it does, but it's not complete. The NMR data requires several files, and sometimes they're not all there, and so on. You can sort of imagine there's lots and lots of things like that. What are the metadata? So in some prior databases of this general flavor, you've had to spend a lot of time saying, oh, it was a 600 megahertz instrument, and the solvent was chloroform, and I acquired it at 25 degrees centigrade, and so on and so on. Actually, all that information is in the metadata file that's with the data. So we don't ask users to do any of that. But in order to extract all those data, we need to be able to do that consistently from those files, even though some of those fields are sort of user serviceable. So like NMR administrators can set the names for the solvent. So it means that even though there's a solvent tag in the file, the name you get isn't always the same name. Sometimes you get different names for the same thing, which obviously makes a mess of the database. So we do a lot of housekeeping. We have a default standard, and then we have a list of known alternatives. And as long as it's a known alternative, we just fix it. And if it's something we've never seen before, then someone has to go look at it and decide what to do about that. Which is, obviously, the more you do it, the rarer that becomes, but you always find some surprising things. Sometimes it's the name of the person doing the work, which is in the solvent box. And then you have to try and figure out. So you can open the spectrum and you can look at it and you can figure out what the solvent is. So there's all sorts of stuff like that. And then there's also all the checks we did with the other structured database. They apply to the NMR database as well. I'm going to run out of time here. If the compound's in there already, what's the embargo status? So people are allowed to deposit ahead of publication so that those data are in the data availability statement and they can be made available to reviewers, but not available to the general public, so that your data aren't released ahead of your paper. So we have to keep track of that embargo status and make sure we don't pre-release things we shouldn't, and so on. And then people always want the accession number. They want the number that goes back in their paper so that they can tell people where to find the data. And so that's something else you have to kind of manage and keep track of. But it is working. So we have had this running now for about, well, however long that is. It's sort of 18 months, but the early years, a lot of that was just like closed. Only our team really knew that it was there until we were sort of testing it and building out extra features and things like this. We have accrued 10,000 NMR spectra, just shy. We're like 9,000 something at the moment. And I put this red line on here. So JMP is the Journal of Natural Products. So this is an ACS journal. It's one of the places where that publishes a lot of new natural product structures. And they were the first journal I know which mandated NMR deposition as part of the deposition process. So if you publish a paper there which reports new natural products, you're now required to put the raw NMR data in the public domain somewhere. And you can't say that there's that it's causative. It's certainly correlative. That the mandate was required, the slope goes up. But the slope may have been going up anyway as the word got out about the database. You can't really tell. But it certainly hasn't hurt the process. And so now we were able to gather about 1,200 raw data with which to first launch the database on the day it went live. And so now we've done sort of 10x that by asking the community to feed in spectra. And we get, it varies a lot, but sometimes as many as 100 spectra a week. Way more spectra than we could get by going and asking friends and family for depositions and to put data in. So, in the long term, I think it's a really good way to do that. But still, the uptake is not as strong as it should be. And I think there's a lot of work to do to convince people that it's for the collective good that we should do it, and that there is some sort of return on that investment for people individually as well, whatever that might be. Okay, I'm clearly not going to tell you about applications today. I'm going to run completely out of time, but that's okay. The last thing I want to tell you is sort of maybe a little bit of a future perspective on what this means for our our project, but also sort of more generally in this field. So we set out to build a database of NMR data for natural products. But what we accidentally built was a system which monitors all the papers about natural products discovery in real time. So that wasn't originally the objective, but by virtue of tracking all the RSS feeds and then watching them and having someone review those papers, there aren't that many papers about natural products every day. So this is all done by one person. But what comes out is this sort of real-time, complete coverage of all the natural products related publications. It's not perfect. If the paper is about something else, but a natural product is also discovered, sometimes we don't catch them. Or if it's in a journal that's not part of our 50, we don't catch them. And we're working on trying to broaden that coverage now. But it's not bad. So at the moment, we look at natural products papers, but actually we're tracking all the papers that are in the natural products arena. There's no reason why we couldn't build another machine learning model which would recognize papers that reported biosynthetic gene clusters or that reported other applications. Applying those other models to the existing database is really not that difficult. And so you can imagine that it could become a place where you might actually track sort of thematic details in real time, not only that those papers matter, which is something you could do with Google Scholar, but also what the contents are, which I think is maybe a little bit more complicated. So at the moment, those data go to NPMRD, but now already we've stopped doing these like large-scale biannual curation blasts with the Natural Products Atlas. Much of the information we need to keep the Atlas maintained is now being generated sort of automatically by running this other database project. It's not perfect. We still need to do some work in that area, but it's definitely an improvement. There's no reason really why we can't also feed those data to other databases like MIBig. At the moment, every time they do a release, I spend a summer reading biosynthesis papers in order to make the connections properly, which is quite time-consuming. So you can imagine that in the future, that's one possibility for sort of federating the data to domain-specific experts, but helping the sort of large-scale curation to be done at a systems level, at a domain-specific level. And then lastly, at the moment, we only collect NMR data. But actually, once you've built the environment where someone can submit the data that goes with their paper, NMR data is comparatively complicated. Encouraging them to submit their MS data at the same time, and then delivering that MS data to their repository of choice is not an unreasonable thing to think about doing. So we definitely do not do this at the moment. It would be a whole nother layer of complexity. But it is tempting to think that you could do them all in one place, which, if you think about deposition fatigue and the people being fed up with having to do this in lots and lots of different places and maintain lots of accounts and things, could be valuable. And we could do that not only for MS data, but we could help MIBig with collecting BGC data and all sorts of stuff like that. So I was wildly optimistic about how much time I would have to talk about the application, so I'll have to leave that for another day. But the last thing I would like to do then is thank all the people who actually do all this work. So there have been an enormous number of collaborators on the Atlas project, and it would have been impossible to do it without them. So we say that if you curate 1,000 compounds, you get your name on the paper about the Atlas. But that's a lot of papers to read. And there are there's other bits of swag. You can get T-shirts and mugs and all sorts of stuff if you hit different metrics. So we're really appreciative to all the people who invested a lot of time in this project on a volunteer basis. And then there are a few folks in my lab who deserve particular mention. So Jeff Van Santen is a developer who built, where did my pointer go? There it is. Well, anyway, you can see him there at the bottom. Jeff Van Santen, he's our developer. He built most of the stacks that we use and prototyped a number of others. And so he's really responsible for a lot of that side. Above him there, Ella Poynton, she's the data scientist that manages the data curation at the moment. And above her is Matt Pin, who is our current developer for the NPMRD stack and built most of that architecture. And then, the rest of the team and these folks on the right for the money to actually do the work. So with that, I'm going to close out and thank you all very much for your attention.